Yo, I hunt people every night. Oh, produced by.
Listen, DNA molecule that goes all over the living things and made of deoxyribose, a fucking rich sugar, and a phosphate group related to phosphoric acid, and then nitrogen is basically ATRCRG. In RNA, there's ribose instead of deoxyribose that completely turns the whole strand into something totally new. Then G comes in and turns into uracil, and maybe people want to do exactly what I do. From my point of view, instead of a double helix, RNA has a single strand. That's exactly what makes it cool. mRNA means genetic info from the DNA to the ribosome, where it gives amino acid sequence. RNA is the power of the ribosome, is essential for protein synthesis in all living things. Whoa, tRNA transfers specific amino acids to growing peptide chains at the ribosomal site. Stupid idiot, I don't think you get it. I'm one of the greatest ever, period. Every syllable is true. We're teaching you since your teacher is long overdue. It should all be a review to all of you fools.
We'll teach you, and we'll learn you a thing or two. And we'll teach you, and we'll finish your biology for you. Well, Mendelian inheritance is a theory of how hereditary characteristics are passed on from F1. Dominant traits show up more often, what we call the rarer traits, and recessive traits because they're really very rare. Homozygous is when both the traits are exactly the same. Heterozygous is when they're both very different. Genotype is two traits, while phenotype is the visual representation of the two traits on a being. The law of segregation is the principle stating that the offspring is one factor from each parent during production. The law of independent assortment is the principle stating that the laws of chance govern which traits their offspring get. The law of dominance is the one factor in the parent dominates the other, while the both of recess. Multiple logates is when there are more than two alleles of abnormally, just like in many blood types too.
Stupid idiot, I don't think you get it. I'm one of the greatest ever, period. Every syllable is true. We're teaching you since your teacher is long overdue. It should all be a review to all of you fools.
Cause we'll teach you, and we'll learn you a thing or two. And we'll teach you, and we'll finish your biology for you. Will.
Co-dominance is when the dominant trait and the recessive trait show up with a heterozygous trait and another allele. Incomplete dominance is when the dominant trait combines with the recessive trait to make the third phenotype. Sex-linked disorders are traits determined by a recessive allele located on the X chromosome of the parent. If the mother has the disorder, then the son's allowed the disorder, but then the daughters will have no disorder since they have two X. If the father has the disorder, then the offspring will be normal, but the daughters will carry the same disorder with them. Transcriptionist is when a segment of the DNA is copied into RNA via enzyme RNA polymerase. Translation is when the RNA is decoded by a ribose complex chain produced from amino acid chain. I'm the kind of guy who stops the microwave in 1 second just to feel like a bomb booster because it's a lot of fun. You stupid idiot, I don't think you get it. I'm one of the greatest ever, period. Every syllable is true. We're teaching you since your teacher is long overdue. It should all be a review to all you fools.
Cause we'll teach you, and we'll learn you a thing or two. And we'll teach you, and we'll finish your biology for you. Well, DNA gyrus makes the whole end a double helix, and each side separates through the replication fork. The leading strand raises towards the replication fork, while the lagging strands goes the opposite direction fork. The helicase combines the DNA strands and proteins, keeps them from merging together because that would be bad. DNA polymerase has nucleotides to the strand. The nucleotides pair with the complementary ones. The complementary ones are on the existing strand. DNA polymerase also proofreads the DNA too. After this, the DNA is put back together using DNA ligase, which is another enzyme. Okazaki fragments are glued together by DNA ligase to make another full strand of DNA. Adenine goes to thymine, while guanine goes to glycine. DNA always goes to in 5 to 3 direction. The special instructions for making a protein are found within the nucleus in the cell's DNA as a form of genes. Through a process, all genes are transferred. The information contained in the DNA is transferred to a molecular RNA. RNA is exported from the nucleus to the cytoplasm with the help of specific protein machinery. Once in the cytoplasm, the RNA is decoded by ribosomes, which use the decoded information to make a protein chain. That's it. Yeah. We out.